Thank you very much. Welcome to you. Hello, everyone, and welcome to this webinar where we will discover the amazing world of arbuscular mycorrhizal fungi and their benefits to crops. Let's start with the outline of the webinar. We will discuss of mycorrhizal symbiosis. We will discover what they are. We will discuss also arbuscular mycorrhizas and the functional diversity of arbuscular mycorrhizal fungi. Then we will treat a very new subject which implies the nutraceutical value of mycorrhizal plants. And then last but not least, we will discuss about Microlysosphere. So let's start from mycorrhizal symbiosis. What are they? They are beneficial symbioses established between soil fungi and plant roots. They involve more than 6,000 fungal species and about 240,000 plant species. So we can claim that in nature, 90% of plant species do not have roots. They have mycorrhizas, from Greek, mycos, fungus, and rhizon, root. They are associations between soil fungi and plant roots. Then you can see in this figure that mycorrhizas are diffused all over the world. The different colors represent different kinds of mycorrhizas, but you can see even if there are different colors, all the kinds of mycorrhizas are diffused worldwide. So just to mention, the main types of mycorrhizas are ectomycorrhizas, ectoendomycorrhizas, endomycorrhizas, and within endomycorrhizas we find helicoid and orchid mycorrhizas and arbuscular mycorrhizas, which are the most important types of symbiosis, of mycorrhizal symbiosis. We already said that they are mutualistic symbiosis. Why? Because mycorrhizal fungi are heterotrophic microorganisms and they must receive the carbon from the host plant. So they receive sugars from the plants, and in exchange they transfer mineral nutrients to the plant. Mineral nutrients which they are able to transfer from the soil to the plant by means of a very large extra-radical network of hyphae. This mycorrhizal mycelium is very important, and it's the key structure for the uptake of nutrients from the soil to the host plant. And in 1997, it was named by the scientific journal Nature the World Wide Web. So let's move forward with the arbuscular mycorrhizas, which I said are the most important types of mycorrhizas. They occur in 80% of land plants, and these plants include the most important food crops, from cereals like rice, corn, barley, wheat, millet, to legumes like beans, lentils, cowpeas, peas, and peanuts, to most vegetables and fruits, including also grapevine and olive. They occur also in many economically valuable plants, such as sunflower, cassava, cotton, sugarcane, tobacco, coffee, tea, cocoa, rubber, and oil palm. Arbuscular mycorrhizal fungi are not host-specific. What does it mean? It means that a fungal species of AMF, as I will define this arbuscular mycorrhizal fungi, this means that a fungal species of AMF can colonize thousands of host plants, and one plant species can host in its roots many different fungal species. So just mention the taxonomical position of these fungi. They belong to Glomeromycota, and so far 334 species have been described. They are also obligate symbionts. This means that they are not able to complete their life cycle without colonizing a host plant. So just have a look to their life cycle. We have here a quiescent spore, which we can find in the soil. The quiescent spore is able to germinate and to form a pre-symbiotic mycelium, which is able to recognize chemical signals which arrive from host plants. Then, when the host plant is recognized by the fungus, it is able to produce peculiar infection structures, which are these ones, called appressoria. Appressoria are able to germinate and to colonize the root. They colonize the root intercellularly and intracellularly. In cortical cells, the intracellular penetration gives rise to peculiar structures called arbuscules. We will see them just now. Arbuscules are very important because they are the key structure of the symbiosis in the roots, and they are the place where the exchange of mineral nutrients and sugar take place in the root. So when the fungus has reached its ecological niche and it's able to absorb sugar, it continues its growth externally. So it forms a large mycorrhizal mycelium, which eventually is able to produce new spores and which will give rise to a new cycle. Just have a look under the microscope to these arbuscules. Here in this picture, you see a piece of root which has been differentially stained just to visualize arbuscules. Arbuscules are these tree-like structures, and this tree-like structure is due to the continuous branching of the penetrating hypha inside a cortical cell. Here you can see a very heavily colonized root where you see some vesicles. Here there are vesicles which are formed after some time after the colonization, and here you see also some spores because some of these arbuscular mycorrhizal fungi are able to produce spores in the roots. So what happens when a plant is mycorrhizal? A mycorrhizal plant usually shows an enhanced growth and fitness and a higher tolerance to biotic and abiotic stresses. And also, it shows a higher efficiency in the mobilization of and transfer of soil nutrients, in particular poorly mobile nutrients like phosphorus, and in water uptake. And here you see some grapevines, and here there are no mycorrhizal plants and mycorrhizal plants, so that you can visualize just this higher efficiency in growth and fitness of the plants. So how does it happen? Here in this figure, you can see a plant root with a yellow zone. This yellow zone is a P depletion zone, which is formed by the fact that the uptake of P by roots is very, very high compared with the P flow in the soil. And what happens when the plant is mycorrhizal? It happens that the extraradical mycelium, this is the intraradical mycelium, just very schematic, and the extraradical mycelium is able to grow beyond the depletion zone and to absorb P from soil sites which are unexplored by the roots. In this other figure, you can see the three interfaces which are important in P uptake. The red one is the soil-plant interface, where plants are able to uptake for phosphorus from the soil. The green one is the soil AMF interface, where arbuscular mycorrhizal fungi are able to absorb P from the soil. And the blue one is the interface inside the roots between AMF and plants, where the exchange of nutrients occurs. And just have a look to this extraradical mycelium. Many studies have demonstrated that phosphorus and nitrogen transporter genes are expressed differentially in this extraradical mycelium, which becomes functionally able to capture very high levels of nitrogen and P from the soil and to transfer them to the host plant. Well, here, 3 years ago, we were able to harvest some of this extraradical mycelium. You see in this very nice picture a mycorrhizal root, this, and you can see also the extraradical mycelium, which is spreading from the colonized root into the nearby environment. We were able to harvest this extraradical mycelium, and we found that ammonium transporter genes are differentially expressed in this extraradical mycelium. And we have demonstrated this with 3 different mycorrhizal fungi, Rhizoglongus irregulare, Phanelliformis mossi, and Phanelliformis coronatus. This extraradical mycelium is very important also from an agricultural point of view, because it has a long-term survival in the soil, and this long-term survival is functional to the establishment of mycorrhizal symbiosis with the following crop. If we have a very highly mycotrophic crop, then we harvest it. Plants die, but ERM, which is extraradical mycelium, doesn't die. Its lifespan is uncoupled from host plant lifespan. What does it mean? Just have a look to this figure. Here we performed a time course experiment, and here you find the time in days, and here the mycelial viability, which is the viability of the extraradical mycelium. We removed the shoot from some plants, and here in the red line you see the results, and here you find the intact plants. And you can see very well that even after 2 months, the dead plants, we can call them, the dead plants had the mycelium of the dead plants had the same viability of the mycelium of intact plants. And this viability slowly decreases in time, but even after 150 days, this viability is quite high, because it's more than 50%, still more than 50%. So this extra-radical mycelium remains in the soil and is able to colonize the new crops. Well, one could ask, what's the length of this extra-radical mycelium which spreads from the roots? Well, the length is about 10 meters per gram of soil, which is a lot. But we were able to reveal its structure in a bidimensional system like this. You can see here in this very nice picture some roots, which are the blue, the dark ones, and then you see the extra-radical mycelium growing from the roots onto the membrane. And the visualization of this intact extra-radical mycelium allowed us to understand that the hyphae, when then they come into contact, like here and here, when they come into contact, they fuse by anastomosis, forming a real network of interconnected hyphae. And this is very important for the transfer of mineral nutrients from the soil to the plant. We were very curious, and the next question for us was just to try to understand whether different mycorrhizal networks were able to fuse when they were spreading from different plants. And we have shown that actually, if you see here, this part, we differentially stained the hyphae, and you can see here the mycorrhizal network of the plant on the left and the mycorrhizal network of the plant on the right. And in this part of the membrane, you see the hyphae which come into contact and fuse. And this happens with plants belonging to different species, genera, and families. So we can claim that there are many below ground plant interconnections which are the result of the connections by means of extra-radical mycelium. These findings of ours opened the way to studies which were aimed at showing whether this interconnected hyphae could be functional to nutrient transfer among plants. And some colleagues of ours in Denmark, using radioactive P32, demonstrated that giving P32 to a donor plant, it was transferred by the hyphae to a receiver plant. So P can be transferred from one plant to another by means of this extra-radical mycelium. So just move forward with functional diversity of arbuscular mycorrhizal fungi. Here we find a morphological diversity. I wanted just to show you how they look like. These are different species of mycorrhizal fungi, and you can see that they form spores which are aggregated in clusters or in sporocarps like here, or may be formed singly at the end of a hyphal tip. They are different colors and different diameters, but I want just to stress that for being microbes, these fungi are very large, because the diameter of these spores range between 50 and 500 microns. So this is quite a diameter for being a microbe. So I want to say that they are very nice, they are different colors, and all their these features have been utilized for taxonomical classification of these fungi. But just let's start with functional diversity. I said that they are functionally diverse. In fact, you can see this picture. These are lettuce plants, and we inoculated these lettuce plants with different arbuscular mycorrhizal fungi. And you can see here that these plants do perform in a very different way from these plants, which are inoculated with different fungi. So fungi are functionally diverse. So they can give different performances to the host plants. They differ for efficiency and for infectivity. So let's just define infectivity and efficiency. Infectivity is the ability to establish rapidly and extensive colonization of plant roots. And this is important when we utilize them in agriculture. And efficiency is the ability to promote plant growth, nutrition, and health. So these are two parameters which are very important for the functional diversity. Obviously, infectivity is much important for the fungi which are in the soil must be very quick to infect, to colonize the plants, and efficiency is important for the nutritional status of the plant. So just have a look to some results we had some years ago. We performed some experiments with different plants, onion, pepper, and lettuce, and we utilized two different AMF treatments. Treatment 2 and treatment 22. Treatment 2 was represented by different fungi from 22. So they positively affected plant growth, and because here we have shoot dry weight, and nutrition, because we have here shoot nitrogen content and shoot phosphorus content. You can see here that for shoot dry weight, onion plants which were inoculated with the treatment 22, pepper and lettuce, 22 pepper, 22 lettuce, showed a statistically different and higher shoot dry weight compared with the plants treated with treatment 2. And so it's the same for shoot nitrogen content and shoot P content only for pepper and lettuce, because for onion we have a larger content of shoot P and N, but they are not statistically different. The statistical differences we found, statistical difference in pepper and lettuce. So if we want to use this AMF in agriculture as biofertilizers or biostimulants, we have to perform studies to find the best combinations between host plants and fungal symbionts. Just to see it with the naked eye, you can see the results of some experiments we have done with olive trees and grapevines. You can see the control plant here, and the plant inoculated with one fungus, and the plant inoculated with another fungus. And you can see very clearly that they differ. So AMF are not all the same. We have to study them and to select the best ones. So just to introduce a very important, interesting, and new innovative research topic, let's talk about nutraceutical value of mycorrhizal plants. We investigated the role of NF in the promotion of nutritional and nutraceutical value of food, and this is very important. I will show you some case studies, starting from artichoke, because artichoke is functional food for itself. And when it is mycorrhizal, the content of phenolics, which are very important for our health, and the anti-radical power increase very much. And just another case study about tomatoes which are produced by mycorrhizal plants. You can see our results, and we have published these results in the British Journal of Nutrition, which is a very important journal. You can see here that the nutritional value of tomato fruits produced by mycorrhizal plants is higher, especially for phosphorus and zinc. But you can see also that we found a higher percentage of lycopene in tomatoes produced by mycorrhizal plants. And lycopene is very important as a secondary metabolite because it has health-promoting properties, important health-promoting properties, and also it has been shown to prevent some kind of cancer. So let's go to another case study which involves basil plants. In this case, the basil we experimented was a red leaf basil called dark opal. I want to stress that this experiment was carried out under commercial growth conditions, and this is important. And we found that AMF increased the production of some health-promoting phytochemicals, such as anthocyanins and rosmarinic acid. And another case study, lettuce, another plant, lettuce, and you can see here that our lettuce was a red leaf lettuce. This red leaf lettuce produced more anthocyanins than controls. Here you find the control, and here the quantity of anthocyanins produced by two mycorrhizal fungi, Arbuscular mycorrhizal fungi. And it's important to stress this fact that one of the two, Rhizoglomus irregulare, was more efficient than the other, which was Phaneliformis mosse, in increasing the levels of the healthy metabolites, in particular anthocyanins. So again, this study showed that AMF are functionally different, and this opens the way to studies on the selection of the best performing strains, also in the case of nutraceutical value of mycorrhizal plants. So last but not least, we will discuss of Microlysosphere. Well, we will start from the nice picture we have already seen. This was the picture of the mycorrhizal root with much extra radical mycelium around. So if we take some of these hyphae singly and we look at them under a high-power microscope, you can see here very nice small yellow spots. What are these yellow spots? Are bacteria which are attached to the hyphae. And if you take a cross section of a hypha and you look at it under the electron microscope, you can see this hypha, which is a little bit leaky because there is some material around, and you see many bacteria associated with the hyphae. And if we have a look to the spores have a complex system of walls, and here you can see that in the outer wall we find attached different bacteria, and here you can see it much better because here there is the cytoplasm. This is the inner wall, the outer wall layer, and attached to the outer wall layer you can see many bacterial cells. So who are they? It's important to know who are they and also what are they doing there. So who are they? We try to identify them by culture independent methods, and I'm showing you the results. We compared the bacterial communities associated with 6 different mycorrhizal fungi, AMF. And just for an example, you see these two. This is the bacterial community of an isolate of Funaliformis mossae from Arizona, and this the same species from Indiana. So they are 2 isolates of the same species, and you can see the difference of the bacterial communities associated. So they are diverse in the diverse species and isolates, and they have no relationship with the taxonomic position of the isolate or of the species of the fungus. And just for an example, if you see the same 2 population of bacteria, this is from the Indiana isolate and this is from the Arizona isolate, you see that they are very different and very distant in APCA. And we could just ask why there are these different differences. We think that this bacterial diversity is due to the fact that different AMF, different arbuscular mycorrhizal fungi, have different metabolic activity. They can exudate different chemical compounds, small amino acids and organic acids, which are good for bacteria. And these compounds can exudate in different quantity and quality, and this can make the difference between the diverse bacterial communities. So what are their functions? Because we would like to know what are they doing there. So if we want to discover the functions of the bacteria, we have to isolate them in pure culture and to do classical microbiological analysis, because we want these bacteria in our hands. So we try to isolate the bacterial communities living in associated with one of the 6 fungi we have studied before. We isolated 374 strains from the spores of this Rhizoglomus, and they had different functions to start because we utilized 4 different selective media, and we found heterotrophic bacteria, but also actinobacteria, chitinolytic bacteria, and nitrogen-fixing bacteria. But we selected 122 to perform the real selection, and we found that they are multifunctional. In fact, we found that 49% of them were able to solubilize mineral P, as you see here, the solubilization. Then we found that 70% of them were able to solubilize organic phosphate in the form of phytate. And you can see here the clear zone. And 66% of them were able to produce siderophore, which are very important for plant defense from pathogens. And we found also that 43% of them were able to produce plant hormones, like, for example, indolacetic acid. So the next question for us was about commercial inoculum, which is utilized by farmers. So are bacteria associated also to commercial inoculum? And we performed some experiments using both culture-dependent and culture-independent analysis. And by using Illumina sequencing, we found 276 bacterial genera, which belong to 165 families, and mostly were represented by Proteobacteria and Bacteroidetes. But the most important thing was the functional part of this study, because we found a very high level of functional bacteria. For example, the nitrogen-fixing bacteria and the bacteria solubilizing phosphate ranged in their numbers from 10 to the 5 to 10 to the 6, which is a very high number. The next question, can we utilize these bacteria? Can utilize these bacteria alone or with their AMF? And so we performed an experiment with some colleagues in Denmark who has the facilities for using radioactive phosphorus. And we found that some of these bacteria are able to facilitate phosphorus uptake in maize. From this picture, you can see the results of some of the experiments we have done. These are non-mycorrhizal maize plants. These are mycorrhizal plants. And you can see that mycorrhizal plants show an increase of 110% in shoot dry weight. But here we have a higher increase when mycorrhizal plants were treated also with their associated bacteria, like Lisinobacillus or like Icynurhizobium TSA41. We had increases of 117 and 127%. And here you can see very, very clearly the use of radioactive P, and what are the results? The counts per second in shoots of radioactive P. You can see very clearly, these are the non-mycorrhizal plants, and these are mycorrhizal plants. But the pink squares represent arbuscular mycorrhizal inoculation alone, and the red and purple squares represent the plants which had been inoculated with AMF and some bacteria like TSA3, CH19 and TSH41. And you see that in some cases the bacteria are very, very much efficient in pea uptake. But in addition to these direct effects on pea mobilization and facilitation of uptake, we found that 7 out of 10 of these bacterial strains significantly improved the growth of extraradical mycelium. And you know how much extraradical mycelium is important for pea nutrition. So now I give you just a graphical abstract of what I have said so far, and you can see very well all the world underground when plants are mycorrhizal and inoculated with plant growth promoting bacteria. And I think that this is a previously unimagined network of interactions which involve plants, mycorrhizal fungi, and their associated bacteria. And I find that it's a complex world, isn't it? And just to conclude, I want to stress this fact. We need more translational studies. These translational studies should focus on the selection of the best performing AMF and bacteria, those which show the most desirable functional activities which are relevant not only for plant growth, nutrition and health, but also for the production of beneficial phytochemicals. Just to conclude, what's, in my opinion, the next challenge in the years to come for agriculture? Bridging the gap between basic science. We do much basic science, but there is a gap between basic science and the formulation of innovative products which could be used for sustainable intensification of food production systems in agriculture. So I thank you very much for your attention. Bye-bye.